Good morning, everyone. And as past president of the Society, let me add my welcome to this second pre-Congress workshop on current progress with PGT. My topic today is a recent study we published on the genetic analysis of the origin of intermediate copy number changes, or mosaic changes as they're often referred to, in multi-cell trophectoderm biopsies identified by NGS-based methods using SNP genotyping and karyomapping to identify meiotic aneuploidies. Preimplantation genetic testing for aneuploidy, or PGT-A, by NGS-based methods is now widely used to select euploid blasts for transfer. Full copy number changes, indicating trisomy or monosomy, are likely to be caused by meiotic segregation errors, particularly maternal age-related errors, as the whole embryo is affected following fertilization. On the other hand, intermediate copy number changes between full trisomy and monosomy may be caused by postzygotic mitotic segregation errors or secondary malsegregation events resulting from spindle abnormalities, which result in chromosome copy number mosaicism among the biopsied cells. And this can occur right up to the blastocyst stage, because we have observations that show that there are abnormal spindles even in the trophectoderm at the blastocyst stage. Now, it's important really to distinguish intermediate copy number changes from full copy number changes, because recent evidence indicates that low level, at least, intermediate copy number changes may be of no clinical significance. And we want to avoid, if possible, rejecting embryos for transfer that actually might be viable, as has been shown by many studies now. So to investigate the origin of full and intermediate copy number changes, we use SNP genotyping of polar bodies, and embryo DNA samples were analyzed by meiomapping and karyomapping and compared to the profiles that result from NGS-based copy number analysis in the multiple trophectoderm cell biopsies. This is an example of a high-resolution NGS-based chromosome copy number plot. The advantage of NGS-based methods over previous methods like array comparative genomic hybridization are that the changes in mapped reads are linearly related to the number of chromosomes in the sample DNA, subject, of course, to whole genome amplification bias and other factors, including software smoothing of the variation between different bins across each chromosome and so on. In this case, there is clearly a trisomy for chromosome 18, but the displacement fails to reach the expected 3 copies, which we define as 100% full copy number change, as indicated by the green line. Also, there are clearly 2 intermediate copy number losses for chromosome 2 and 9, but these displacements are only a fraction of that expected for a full monosomy, which would be indicated by the lower, what should be red, but in this particular profile looks a grayish line at 1 copy. Now, various labs have applied different thresholds to distinguish between variation around 2 copies, as we can see here, and full changes of 3 copies or only 1 copy. There was a general consensus until recently that the thresholds should be set at 30 to 70% of the expected full copy number changes, and this is what I've represented here by the red lines, so that you can see that the variation around 2 copies falls within the two 30% boundaries, whereas the other displacements are either on the line or beyond the line. So in this example, the trisomy 16 is likely to be reported as a full trisomy because it is beyond or greater than 70%, whereas the two losses are borderline intermediate changes at around 30%. And so that really shows you the difficulty of, do you call those as mosaic changes, or are they only a result of noise? It clearly looks, because there's a step change from 2 copies, as though there's definitely some kind of aneuploidy in the sample. So we use these thresholds, 30 and 70%, as a starting point for our analysis. Now, meiotic errors are inherited through the gametes following meiotic errors in meiosis, and they're inherited through the gametes at fertilization, predominantly female meiosis. And because of that, they affect all the cells of the embryo, or would be expected to. So they'd be expected to result in a full copy number change in the trophectoderm, even at the blastocyst stage. To test this, we identified meiotic errors through polar body analysis for maternal errors and karyomapping of DNA amplified from the trophectoderm biopsies at the blastocyst stage, as seen here in this example. This then allowed us to measure the corresponding copy number changes following NGS-based copy number analysis with the same amplified DNA samples from the trophectoderm biopsies. What you can see here is karyomapping viewing software that I created some years ago, which displays genome-wide across each chromosome the parental haplotypes present and the position of recombinations or crossovers. Furthermore, it highlights any regions where both haplotypes from one parent are present, indicating meiotic trisomy. And you can see various examples of that, particularly here, for example, where instead of having a single chromosome with recombinant regions of the different haplotypes, we have both maternal haplotypes present over this region of the chromosome on the short arm and this region of the chromosome on the long arm, indicating the presence of 2 distinct maternal chromosomes due to a meiotic error. In this example, if you look carefully, there are no less than 4 meiotic trisomies and 1 monosomy. So if we now look at the corresponding NGS-based copy number profile for samples like the one I've just shown, in this case I'm showing 2 examples of trophectoderm biopsy, the upper 2 panels, and 2 examples of arrested embryos in the lower panels, we can now identify which of the copy number changes we see in the profile are due to meiotic changes and which are presumably of mitotic origin, because there is no evidence at the molecular level that there are two paternal or maternal chromosomes present to cause the trisomy. So what I've done is to indicate, using the red arrows here, where there was evidence that the change was meiotic. So for meiotic trisomies, and this example here, this example, and this example, there was clear evidence for two maternal haplotypes in regions of these chromosomes. In the case of losses, indicated here, it turns out that SNP genotyping and karyomap analysis is quite sensitive. So we can't be sure that these are meiotic rather than mitotic, but it's quite likely because we saw no evidence for the missing chromosome. There was no SNP evidence showing crossover, a pattern of crossovers, that actually that a minority of cells in the biopsy were indeed had the potentially missing chromosome. So we assume that these losses are meiotic and so on. But as you can see, particularly in this sample, there are other quite clearly full chromosome gains for other chromosomes which showed no evidence of meiotic trisomy and are presumed to be of mitotic origin. So I'm now going to focus on just the middle two panels here. And these are the results from 7 trophectoderm biopsies in which there were 38 whole chromosome copy number changes, which when we looked at the karyomap patterns, we could see evidence for meiotic changes in 13 of these changes, but in the other 25 they were all mitotic. So already this is quite interesting information because there are multiple abnormalities in these trophectoderm samples. The patients, a couple of them were advanced maternal age, but not all of them in this case. So nevertheless, the number of mitotic changes picked up by copy number profiling by NGS was far in excess of the number of meiotic aneuploidies. And of course, it's the meiotic aneuploidies that we're worried about, which we know will affect the whole embryo and we know are lethal. So now we were able to measure separately the displacements for the meiotic changes and the mitotic changes, and those are displayed here. If we now superimpose our threshold of 30 and 70%, you can see that all of the meiotic changes, apart from one, would have qualified as full changes and therefore would have been reported as trisomies or monosomies. Conversely, with the mitotic changes, some of them fell below 30% and would not have been reported at all as being possibly a result of noise around 2 copies. Many of them are indeed between 30 and 70%, but 4 of the 25 were above 70%, and some of them were very much higher than 70%. The explanation for this is not clear, but it may be that these mitotic changes occurred early in cleavage, and therefore, in the same way that meiotic aneuploidies affect all the cells of the embryo, perhaps these changes were introduced after fertilization or during fertilization at syngamy, and there have been various papers that have shown that there can be multiple losses at that stage, and have resulted in this situation where you get what appears to be 100% copy number changes or more in the trophectoderm biopsies. So this requires further investigation and is interesting. Now, if, as some groups have suggested, we change the goalposts, if you like, the thresholds for identifying intermediate changes, and place the upper threshold at 50% and not 70%, then the effect of doing that in this data set is that we do indeed now pick up all of the meiotic changes, but we pick up a greater number of mitotic changes as full copy number changes. And the problem with that is that we don't know the clinical significance of these changes because they may not affect all of the cells of the embryo. They clearly are perhaps form a majority of cells in the trophectoderm cells that were biopsied, but these could be a result of de novo errors in mitotic segregation within the trophectoderm, and I've already said that we have seen spindle abnormalities in the trophectoderm at the blastocyst stage, and so may be of no clinical significance and may simply be cell lethal and be eliminated at later stages. So interestingly, in our clinical practice, in an IVF program dedicated to single frozen blastocyst transfer with optional aneuploidy testing, we routinely reanalyzed embryos that only had intermediate copy number changes to see if they were genuinely aneuploid with meiotic aneuploidies, or only had mitotic changes of unknown clinical significance, so that following genetic counseling, the patient could opt to have those embryos transferred. And I'm just going to concentrate on the top part of this table with 11 samples that were reported as normal, all with just mosaic changes for these chromosomes, and in some cases for parts of chromosomes. But we're going to focus mainly on the whole chromosomal aneuploidy in these samples. So this was the NGS result as originally reported. We then did karyomapping, so we had to genotype the two parents to do this. And then we reran the same amplified DNA that had been used for NGS copy number analysis. And you can do this very effectively, even though the normal amplification, whole genome amplification protocol is different for NGS and karyomapping. And what you can see among the 11 samples is that 4 out of these samples were identified by karyomapping as euploid. So if we look on the right-hand side of the table here, 1, 2, 3, 4 were identified as euploid, with no sign of even a partial loss, which can also be picked up by SNP analysis and karyomapping. 2 of the samples were maternal triploid and had extra maternal chromosomes, which could be detected by karyomapping across all chromosomes, and were therefore probably a result of abnormal fertilization and retention of the second polar body. And finally, there were genuine maternal, as it turns out in each case, meiotic aneuploidies for the same chromosomes that had been identified as mosaic by NGS in 5 samples, 1, 2, 3, 4, 5. So this is a very difficult situation because it means that there are limitations, at least at this stage, and these samples go back to NGS-based analysis 3 or 4 years ago, and the techniques have improved, including the introduction of artificial intelligence and so on. But nevertheless, the biology dictates that there will be full meiotic changes, but also mitotic changes with intermediate copy numbers. And it may be impossible to discriminate completely so that some genuinely aneuploid embryos only give you a mosaic result, and some euploid embryos actually give you intermediate copy number results. Finally, we had one very interesting example of what could be misinterpreted as self-correction. So here is the copy number profile for a whole arrested blastocyst. And above it, we have the karyomapping pattern using the same visualization software that I showed you before for the first polar body, the second polar body, and the embryo. Now for the first polar body, during normal meiosis, you would expect one bivalent to be retained in the first polar body and the other bivalent to segregate into the oocyte prior to the second meiotic division. And because of recombination, you would expect to see regions by karyomapping which have both maternal haplotypes. And indeed you do on all of the first polar bodies for these highlighted chromosomes, only some of the 24 chromosomes here, except one. Here you just have a single chromatid, already indicating that there's been an abnormal segregation, leaving only one chromatid in the first polar body. And this would be typical of, for example, premature sister chromatid separation. Now in the second polar body, if we just focus on chromosome 4, which is segregating chromosomes during meiosis normally, we have a single copy of a chromatid in the second polar body and a single copy in the zygote, along with, obviously, since this is an embryo which underwent fertilization, although it arrested, we have also the single paternal chromosome indicated here in red and blue. And actually, if you look very closely, you can see that the segregation pattern is normal because around the centromere, which is indicated by the dot here, by the little gap here in the profile, we have the yellow bivalent retained in the first polar body and green chromatids in the second, exactly as you would normally expect. But of course, in these other chromosomes, we have abnormal segregations of different kinds, leading to trisomies, no less than 4, I think, trisomies in this example, and one monosomy in which there's no maternal chromosome 21. So if I indicate on this copy number profile which are the meiotic changes, you can see that there's considerable variation in the displacement, and it may be partly because there are so many changes in this sample that the software has a problem about deciding where copy number 2 should actually be. So you can see all of these changes. Now, one of them is very interesting because for chromosome 10, the copy number in the whole arrested blastocyst is actually exactly 2 copies according to the copy number profile. And yet there is very clear evidence, both in the polar bodies and in the same DNA sample, that actually there are 2 maternal copies present in some of the cells in this embryo. So in the inner cell mass, it may well have inherited the meiotic trisomy, in which case this embryo would not have been viable. So in summary then, NGS-based PGT-A broadly discriminated between full changes, many of meiotic origin, and intermediate or mosaic changes of presumed mitotic origin. And of course, in recent years, there have been improvements since we analyzed this data in discrimination, but I would argue that it is still going to be difficult to discriminate 100% between meiotic aneuploidies of meiotic origin and those of mitotic origin. And the importance clinically there is meiotic origin embryos. There's very little evidence for self-correction of those, and those are the ones that we want to avoid transferring. At a threshold of 70% or more, almost all of the meiotic aneuploidies were classified as full aneuploidies. However, some mitotic aneuploidies were also classified as full aneuploidies. Only 1 maternal meiotic trisomy was not detected as a copy number change by NGS-based testing in a single arrested blastocyst sample. All of the other meiotic aneuploidies, maternal aneuploidies, detected in the polar bodies did result in copy number changes in the trophectoderm, all of which, barring 1, were classified as greater than 70%. And finally, in our data set at least, we see no evidence for so-called self-correction of meiotic aneuploidies at the blastocyst stage. So the importance of pre-implantation genetic testing for aneuploidy, I think, is reinforced by our data. Thank you very much for listening, and if you have any questions, please direct them to this email on which directs your queries to my website, and I'll try and answer them. Thank you very much.